So we'll work with the audio, okay? Keep the class mode. I hope I'm audible clearly, right?
Yes, sir.
Okay, very well. So tell me, can you see the screen that I'm sharing, both of you?
Yes, sir.
Any doubts? Both of you were there in the last class or no?
I was not there, sir.
Afia?
No, sir.
No? Okay. So actually in the last class, we have finished the chapter 5th, which is the Mendel's genetics, the first chapter of the genetics, okay? And I have not started like something as such, but I just gave a brief introduction of the chapter 6 that we are going to start today, okay? So let's start from chapter 6 today in today's class, okay? Saifullah and Wafia from the beginning. And if you have any doubts regarding anything with respect to genetic disorders or pedigree analysis in the last chapter, you come up to me with your doubts tomorrow. How about that? Okay?
Okay, sir.
Okay, very well. You can also, I hope you both have asked for the recorded lecture link.
Yes, sir.
Okay. So we have started just with the chapter 6. So let me give you a brief recap of what we did. We didn't do much. It was just the beginning. So in chapter 6, we will learn about the molecular basis of inheritance. So whatever we have studied till now in chapter 5, where we talked about genes and alleles, characters, Mendelian laws of inheritance, the exceptions of it, mutation, sex determination, pedigree analysis, and genetic disorders, both on Mendelian, which is the genes and allele mutation based disorders, or non-Mendelian, which is the chromosomal disorders. In this chapter, we are going to understand what is the molecular basis of inheritance. Like when you go inside the cell, what happens inside the cell, which causes all that variation or diseases or inheritance that we studied in the last chapter. Okay. So I started with the simple concepts first. So do you know about DNA and RNA already? So tell me something about DNA and RNA that you know already, Wafi and Saifullah. Let's start with Saifullah. Saifullah, what do you know about the DNA? You can take the help from the board as well.
It's a double helix structure.
And what do we mean by helix? It's
spherical, like spiral.
Spiral. It's coiled. Helical means it's coiled. Yes. And
it is a source of like a transferring of genes.
Yes, it is the genetic material in our body. Correct. And what do you know about RNA? What is RNA?
It is single-stranded or double-stranded. It is made up of DNA, like.
Yeah, so RNA is like normally single-stranded. The double-stranded dsRNA, the double-stranded RNA, we are not talking about here. So for the sake of like to keep the discussion simple, as we'll go forward, I'll tell you about single-stranded and double-stranded RNA both. But let's now figure out that it's single. Okay, first we will discuss that it is a single-stranded. Okay.
Okay, sir.
Yeah. Continue. What else do you know about the RNA?
It is a segment of DNA. Like
Okay,
it is
so
made
we
from
have
a segment of DNA.
Yes, right. It is transcribed from one particular segment of DNA. You're correct. We have Reem. Hello, Reem. How are you?
I'm good, sir.
Good to see you. You were there in the last class, right?
Yeah, I guess.
Yes, you guess you were. You were there.
Last week we didn't have any class.
No, before that, like when we have the last class
Yeah,
where
yeah,
we
I
just
was there.
started the. Yes, right. So I was just giving a brief recap of chapter 6 that we are about to start, and we talked about DNA and RNA. So DNA is a double helix structure. It's called deoxyribonucleic acid, DNA, and RNA is ribonucleic acid. So this deoxy part is not present in the RNA. DNA is double helical. There are two complementary strands that are bound together, and in these strands, basically there are four nitrogenous bases: A, T, G, and C: adenine, thymine, guanine, cytosine. A and G are called purines. I told you this, I was discussing this. And T and C are called pyrimidines. There is another one which is called U for uracil. It is also a nitrogenous base, but uracil is only present in mRNA. Not in DNA, okay? So uracil is present in mRNA, and thymine in the place of thymine. In RNA, in place of thymine, in place of T, okay? Thymine. Just note this down. So A, T, G, C, and U. So A binds with T, purine, with a double bond, double hydrogen bond. G binds with C, okay, pyrimidine. So this we discussed, just the basics, how the binding happens. RNA is single stranded, so the bases are not bound to any complementary strand with another base, so they stay open. And then we discuss this central dogma of biology, which you did this, Reem, with me? You were there?
Yes, sir.
Yes. So would you like to please tell about the central dogma of biology? The last thing.
So I didn't revise.
Yeah, just let's go with the flow. I'll help you. So central dogma of biology is a flow chart, basically, that defines the, write down, central dogma of biology tells you about the genetic flow or flow of genetic information. Flow of genetic information. Okay. So it tells you that inside our cell, what is the genetic material that we have? DNA, right? Yes, Dream. And then what
Yes, sir,
happens?
yeah.
Yes. And
It
what is this?
replicates.
Yes. So DNA can produce DNA. It is self-replicate. It can self-replicate to produce more DNA through the process of replication. Is it clear, everyone?
Yes, sir.
And DNA then forms RNA, just like Saifullah was saying, that RNA is actually formed from a segment of DNA, where a part of DNA is read and RNA is formed, and that process is called transcription. So the gene flow goes from DNA, genetic information flow goes from DNA to RNA, and that finally that RNA can be read by ribosomes. Ribosomes are cell organelles which are present both in prokaryote and eukaryotic cell, right? Class 11th, you must be remembering this. They are also called as the protein synthesis factory of the cell, correct?
Yes, sir.
RNA gives rise to protein, and this process is called translation. So there are three processes that we have to read and understand: replication, transcription, and translation. So this is called the central dogma of biology, the flow of genetic information. Now, I told you that viruses are an exception here because they have the ability, some of the viruses can convert DNA, can make DNA from RNA. Now, if DNA to RNA is called transcription, then RNA to DNA will be called reverse transcription. So they are capable of doing reverse transcription. Okay. At the end, finally, protein is the structural and functional biomolecule inside every cell. So this needs to be formed to carry out any function. Yes or no? All the characters, yes, traits, everything comes from the protein. So everyone understood? Please note this down. Wafia and Saifullah. And let me know when you're done, then I'll start with the chapter. Done, Saifullah and Wafia?
Yes, sir.
Yes, sir.
Okay, cool. So in today's class, we will start with the structure. Basically, we'll talk about the structure. I gave you a homework, but for Reem, did you go back and look at the structure of the DNA in packaging?
Yes, sir, I did.
You did? Okay, so
Yeah.
then you will be able to help me with the discussion. So let's start with the DNA structure. Now, to keep it simple, DNA is made up of small monomers. So it's a long polypeptide, it's a long polymer, right? DNA is a long thread. It's a long polymer, correct, everyone? And if it is a
Yes, sir,
polymer,
yeah.
it must be having monomers. Like when you see a chain, so you must have seen that chains are like this, correct? There are monomers that link together to form a big chain, right? Any chain that we see, it has monomers.
Yes, sir.
Each one of this is a monomer and together they form a polymer. Similarly, DNA also has monomers. So what is the structure of DNA? DNA has basically three things. Pentose sugar, like a 5-carbon sugar. In case of RNA, it's called ribose sugar, and in case of DNA, it's called deoxyribose sugar. That is where the name comes from, deoxyribonucleic acid and ribonucleic acid. So DNA structure has three things. First is a phosphate group, a nitrogenous base. Okay, and that nitrogenous base, you know that there are 4 types: A, T, G, and C. So either A or T or G or C. And depending on, because there are 4 nitrogenous bases, so there can be 4 different types of monomers, right? Each carrying 1 base. The second thing it has is a pentose sugar. The word pentose comes from pent means 5, right?
Yes, sir.
It's a
Yes, sir.
5-carbon sugar. And in case of RNA, it's called ribose sugar. And in case of DNA, it's called deoxyribose sugar. Okay. The third thing it has is a phosphate group. Okay. These 3 together make that one monomer that we were talking about. Understand? And when all these 3 are present together, that monomer is called nucleotide. Okay. So nucleotide is a monomer of DNA. Is it clear, everyone?
Yes, sir. Yes.
Let's go to the structure, how it is arranged. So DNA has like a 5' end and a 3' end. So it has 2 ends. So starting from the 5' end, there is a phosphate group. Okay, I'm writing it as P. I'm not making the whole group. This group is attached to the carbon of the pentose sugar. Okay, now this carbon will have hydrogen. On this side, it will be attached to. So this is the structure that this sugar has. Okay. and this sugar has a base protruding towards inside. Now this, let's say, could be any base. Let's start with A. Okay, so this is one nucleotide. You understand? A 5' phosphate group. Okay, one pentose sugar and one nitrogenous base. Is it clear, everyone?
Yes,
This is
sir.
one nucleotide, one monomer. Now it keeps on forming like this. So from here, it will be attached to another phosphate group, which will again be attached to carbon molecule, which will be showing the same kind of sugar, and there will be a base. Let's say this time it is thymine. Okay, so this is another monomer. You understand?
Yes,
And
sir.
this thing continues. So this time there will be another phosphate, carbon. So just when you are practicing with me, practice the structure as well. It's good that you know it. If you know it, and then there'll be a base. Let's say this time it's G, and so on. You understand? So it will continue. And this one thing which I'm marking here, right? This is one unit which is repeating, right? Basically.
Yes.
Yeah. So this is known as nucleotide. Now remember, see in this, this is like I'm making two strands of DNA, right? I'm not coiling it. Let's say I have unwound it. So in the DNA, a DNA is like this, correct? Yes, everyone?
Yes, sir.
Yes, sir.
So this is one strand, okay, which is represented here. This is another strand which I have not shown. And here is the base, right? These bases are here from one side. From other side also bases come and they join with hydrogen bonding. So this is the base. You understand? So this is just one strand that I have made. So if you see, this is the backbone. Phosphate sugar, phosphate sugar, phosphate sugar is the backbone, right? The strand,
Yes, sir.
two backbones. And what comes towards inside is the nitrogenous base, right?
Yes, sir.
So write down the backbone of the DNA, the backbone of the DNA and same holds true for RNA also. The backbone of the DNA is made up of phosphate group, and pentose sugar. Okay, everyone? This is the backbone. And next line, and nitrogenous bases project inwards from the backbone. Understood?
Yes, sir.
Yes, sir.
So there is another word which is in your book called nucleoside. Okay, so this all three together, phosphate, pentose sugar, and nitrogenous base, is called nucleotide. And nucleoside is just this structure. So what I'm marking in green will be nucleoside. The sugar with the nitrogenous base, where phosphate is not a part of it. You understand? Because this projects towards the side. So this is called nucleoside. How I remember it is, nucleotide is the monomer of DNA and nucleoside is what projects sidewards. So sugar attached with the base makes this sideward projection, right? So you will remember nucleoside is just pentose sugar with the nitrogenous base, and nucleotide is all the three components together which form one monomer.
Yes.
Yes.
Okay. Okay. Now this phosphate is linked to this sugar. Okay. So toward this side is the 5' end of the DNA, and towards this side is the 3' end of the DNA. So there has to be a like direction of the DNA. So this phosphate binds to this carbon of the sugar. So this linkage here, if you can see this linkage, this linkage. So I'm just take sugar. And you see this linkage here, everyone? This is called phosphoester linkage. Phosphoester linkage. Is it clear? And because it
Yes,
is
sir.
on both the sides, one phosphate binds to the sugar on each side, right? Here also and this side also. This side also it is bound to the chain, sugar only, right?
Yes, sir.
Yes. Yes. So two nucleotides are bound. So if you see this one and if you see this other, so they are bound together through phosphodiester. So this linkage through which two nucleotides are bound together are called phosphodiester linkage. Phosphoester and phosphodiester. Will you remember this? Just write it down. Phosphodiester, because 1 and 2. Two things are linking together, right? One monomer, other monomer. That's called phosphodiester. So that's why you can make a unlimited, like a long chain of nucleotides together, because you just have to arrange the all the components of nucleotide and make a phosphodiester linkage between the two. Is it clear?
Yes, sir.
Yes.
Very good. So please make this in your notebooks.
Sir, what is 5-phosphate and 3-phosphate ends?
5-phosphate and 3-phosphate. It's just the 5' means towards the 5'. So this is a 5' phosphate. If you go towards this side, this phosphate with respect to this nucleotide is a 3' end. So if I take this as nucleotide 1, okay, then this is nucleotide 2, nucleotide 3. So nucleotide 1 has a phosphate which is on the 5' towards the 5' end of the DNA, right? But if I talk about this phosphate with respect to the first nucleotide, it is towards the 3' end, right? So this is the 5' phosphate and 3' phosphate for this nucleotide. Do you understand?
Okay.
If you take this sugar as a base, let's say this sugar, the sugar number 1 here is the base. So for this sugar, there are two phosphates that are binding to it. One is this one through this carbon, and the other is this one through this carbon. Here also there will be a carbon, right?
Yes, sir.
So this will become the 3' phosphate for this sugar, and this will become the 5' phosphate for this sugar. Is it clear?
Okay, sir.
So the 5' phosphate binds with the sugar through phosphoester linkage. And the 3' phosphate binds with the sugar with a phosphodiester linkage. Is it more clear now, Saifullah?
Okay, sir.
Please draw this and let me know when you're done. Also note down that if the one strand has a direction 5' to 3', sorry. Okay, then the other strand will have a direction of 3' to 5'. You understand? It's complementary to each other. Okay. Then only they can bind together, no? Just like as a kid, have you ever tried two magnets sticking together? They only stick in one orientation.
Yes.
Yeah. Let me know when you're done, everyone.
Done, sir.
Done. Okay. Others?
Done,
Yes,
sir.
sir.
Everyone done? This
Yes,
diagram
sir.
is clear to you? Because if you understand this, then you will have no problem writing about the structure of DNA. Nucleotide is the monomer, nucleoside is clear, phosphoester linkage, phosphodiester linkage, everything is clear, right? 3',
Yes,
5'.
sir.
Yes.
Okay. Now let me tell you about, so how was this structure figured out? Okay, so some things. So write down, let me dictate you certain points. So DNA is acidic, you know that? That's why its name is deoxyribonucleic acid. So DNA is acidic in nature and is negatively charged due to the phosphate group. So this is one property of DNA you should know. It is acidic in nature and is negatively charged. So this acidic substance that is present in the nucleus was first identified by Frederick Griffith. So remember this name, it can be asked. Frederick Griffith. So he for the first time discovered that an acidic substance is present inside the nucleus. And he called it nuclein. He did not know the nature of it. He did not know what chemical molecule it is, what is the chemical composition of it, but he knew that it is acidic and it is present inside the nucleus. Okay. And this he did very early in 1869. Okay, everyone?
Yes.
After that, there were two scientists. So this is the first point, the second point. There were two scientists called Rosalind Franklin she along with Morris Wilkins. They both conducted some experiments. Okay. Conducted X-ray diffraction experiments. So they diffracted X-rays through the DNA to figure out its structure. So they conducted the X-ray diffraction experiment and collected the data. But they could not understand the data. They did not know what to figure out from this data and what would be the shape of the DNA from this data. Okay. So you can write down, but couldn't explain the data. Taking the advantage of their data, then came, this is the timeline that I'm telling, then came two more scientists, James Watson and Francis Crick. They're called Watson and Crick together. They took the Rosalind Franklin and Morris Wilkins data, took the data and proposed, they understood the data, they spent time on the data, tried to figure it out, and proposed the double helix structure. Double helical structure of DNA. And this happened very late, a long time after Miescher figured out nuclein in 1958. It took a lot of time for scientists to figure out the structure of the DNA. Is this clear?
Yes,
Yes,
sir.
sir.
So DNA is acidic, negatively charged. First it was found. Then Miescher said that this acidic substance is present inside the nucleus and called it nuclein. He did not know what to call it, what was the structure. Rosalind Franklin and Morris Wilkins conducted some X-ray diffraction experiment and collected data, but could not explain the data. James Watson and Crick took the same data, which was collected by Franklin and Wilkins, and proposed the double helix structure of the DNA. And they were given the Nobel Prize for this, to figure out the structure of the DNA. And hence comes what was their theory? What did they propose? So write it as a question. So this question often comes in school examinations and boards. Describe the salient features. Describe the salient features or defining features or characteristic features. They can use any word. Describe the salient features of the double helix the double helix structure of DNA. Written the question?
Yes, sir.
Yep. Now the first thing is what you have already understood above. There are two strands, right? There are two chains. So write down. It is made up of two polynucleotide chains. Two polynucleotide chains where the backbone is made up of sugar and phosphate, right? Remember above? So the backbone is sugar phosphate and the bases, which are the nitrogenous bases, project towards inside. Is it clear, people? The first thing that they said. The same thing that I've shown you here, right? Here. You understand,
Yes, sir.
everyone?
Yes, sir.
Now they come to the polarity. So write down. The two chains are anti-parallel to each other. The two chains are anti-parallel to each other. Okay. So this simply means that if one chain is in 5' to 3' direction, then the other chain will be opposite, 5' to 3' direction. You understand?
Yes, sir.
Yes, sir.
So they are anti-parallel. And similarly, like this arrangement, so you have made it from 5' here and 3' here. So if you pair it with another strand, which is, so what I have shown you here is just the half strand, right, in the figure. If you want to make the second strand also, so there will be a double bond, triple bond here, then another base, and this polarity will change. Now sugar will be on this side, right, and a phosphate, just a image, mirror image. You understand, people?
Yes, sir.
Yeah. Third thing that they said was about the bases. So write down. The bases are paired. The bases in opposite strands. The bases in opposite strands are paired through hydrogen bonding. Through hydrogen bonding. Have you studied about bondings in chemistry now? You must have, right? Covalent bonding, hydrogen bonding, ionic bonding. Yes? People? Yes or no?
Yes, sir.
Okay. So the bases here are paired through hydrogen bonding. Now you have to remember here that adenine forms a double bond with thymine. A forms double bond with T, which I have told you above also. And guanine forms a triple bond with cytosine. Okay. And also to know, adenine and guanine are purines, and thymine and cytosine are pyrimidines. So if you see one purine base pairs with one pyrimidine, but it's not exchangeable. Adenine as a purine cannot bind with cytosine. These are specific. Adenine always binds with thymine and guanine always binds with cytosine. Is it clear?
Yes, sir.
Yeah.
Yes, sir.
And then they told that, so this is about the base. It looks like a ladder. But then in the end, they also said that that's not enough. After this whole thing is done, the two chains are coiled in a right-handed fashion. The two chains or strands are coiled in a right-handed fashion. It's like you take a ladder, hold it from top and the bottom and coil it in a right-handed fashion. Right-handed fashion is like clockwise. You understand?
Yes, sir.
Yeah. And let me draw this coiling for you. So this is what they figure out, right? Can you see this coiling? Now, this part here, one complete turn, is called a pitch of DNA. Okay. And in this one pitch, there will be around 10 base pair. Okay. You understand, everyone?
Yes, sir.
The size, this one pitch of the helix is around 3.4 nanometers, this length. And if there are 10 bases, okay, 10 base pair, then what is the distance between two adjacent bases? If one pitch contains 10 base pairs and this is 3.4 nanometer, then what will be this distance between the two bases, like 1 and 2, this one? 0.34 nanometers. Yes. Yes or no?
Yes.
Yes, sir.
So this is calculated. Rough estimate. So write down. Pitch is this. And two bases are 0.3 nanometers apart from each other. Is it clear, everyone? Understood?
Yes, sir.
Yes, sir.
And then the fifth feature which he told is that the plane, if you think about a plane, if you look at a DNA from the top, so let's say this is one base pair, then it will keep rotating. Do you understand why it is rotating? Because it's one on the top. It's like, have you seen those staircases which are round, coiled, and they go? So you start, think of this as a staircase. Understand? So that staircase actually winds on each other. on itself, like this. You understand?
Yes,
Something
sir.
goes like this. So if you see from the top or from the bottom, you will see that all the stairs are like imposed on each other. You will like a spoke of wheels, right?
Yes, sir.
Yeah. So write down the fifth one was the plane of one base pair, the plane of one base pair stacks over the other. Stacks over the other in a double helix. So this is exactly how it goes. The DNA it keeps on going up and up. This is how it is called. You understand? So if someone sees from the top, all he will see is a round thing with multiple base pair stacks, right? Yes or no?
Yes, sir. Right on. This stacking along with the hydrogen bonding makes the DNA more stable. Do you understand? Now this thing is not present in RNA, right? RNA does not have hydrogen bonding because it's single stranded, and the stacking plane is also not there. Is it clear, everyone? Yes, sir.
Yes.
Please finish writing all the 5 points.
Yes, sir.
Is the structure of DNA clear to you now? Anything, any confusion, please ask. If you have any issue with the structure of DNA, please ask right now. Go through the 5 salient points. I give you 2 minutes because next is packaging. If you do not understand the structure, it will be difficult to understand the packaging. So I give you 2 minutes, go through all the 5 points and let me know if there is any doubt. Time starts now. I'll be back in a minute, okay? Reem, get myself water. Okay, people?
Okay, sir.
Okay, I'm back, people. Done. All the 5 points are clear, everyone.
Yes, sir.
Yes.
Okay. Good. So let me now start with the structure of DNA. Sorry, packaging of DNA. Oh, sorry. Choose. Now, what do we mean by packaging of DNA? First, we have to understand that. Why is there a need of packaging the DNA? DNA, I have been saying this, it's a long polymer, but how long can it be? Any idea? How long, how many DNA threads do we have in one cell?
Only one.
No. How many chromosomes do we have in one cell?
23 pairs.
So total chromosomes?
46.
46 chromosomes, right? So how many DNA threads are there? 46.
46.
Yes. One chromosome is a condensed form of one DNA thread, right?
Yes.
So now let's do some mathematics here. So we know that the distance between two consecutive base pairs is how much?
0.34 nanometer.
Yes. Distance between two consecutive base pairs is equal to 0.34 nanometer. And how much is a nanometer? If I write it in 0.34 mathematical expression into 10 raised to power. How much is a nanometer? If I convert it in meters.
-6.
That's micrometer. Nanometer is -9. Okay. -6
Okay.
meters is micrometer. -9 is nanometers. -12 is picometer. Then ferrometer, like that. So 0.34 into 10 to the power -9 is the distance between two bases. Okay. Now, you know how many total base pairs do we have?
4.
Total base pairs. 4 is the total bases that we have. Total base pairs in
2.
entire all the DNA. Oh, you understand? We have 10 base pair in one pitch. We have 10 base pair here. 10. So if the DNA keeps running, if you keep counting 10, 20, 30, 40, how many total base pairs do we have? This is what I'm asking. Total base pair, write down total base pair in a mammalian cell. In a mammal cell. Approximately true for humans. So that is 6.6 into 10 raised to power 9 base pairs. Quite a lot, right? And this is for a diploid cell. If you talk for a haploid cell, it will be 3.3 into 10 to the power 9. Okay. Now, if you multiply both the total number of base pair and the distance between the two. So if there are this many bases and this is the distance between the two, then you multiply the two to get the total distance, total length of the DNA. So can someone tell me what is the total length of the DNA coming? In one cell diploid. 6.6 into 10 to the power 9 into 0.34 into 10 to the power -9. So this will cancel out. Yes. Yes or no? 10 to the power 9, 10 to the power minus 9.
Yes, sir.
Yes, sir.
6.6 into 0.34. How much? 6.6 into 0.34. Quick. This is around 1/3, right? 0.33 is 1/3. So 1/3 of 6.2 is 2.2. And the unit is meter. 2.2 meters. In one cell. Can you imagine? A cell is not visible with the naked eye. It is such a small thing. It's at the scale of micrometers, right? One cell. And in a space of something which is on the scale of micrometers, you need to keep something, the total length of which is 2.2 meters. How are you going to do it?
By
There's
coiling it.
only the coiling will not work. You have to supercoil the DNA, right?
Yes,
Only
sir.
supercoiling can allow to keep. So you have to keep coiling and packing to an extent where there are multiple layers of packaging, and it becomes so tiny that it can be kept in a small space. For example, the same thing I told you about a thread. So we have a spool over which we keep binding the thread, right? We keep what do you say? The thread is wrapped around. Keep coiling the thread. So maybe this thread is 500 meters long, right? But when you keep coiling it over this small space over and over again, that's called supercoiling. Do you understand?
Yes, sir.
Yes, sir.
So this kind of supercoiling is required. That comes under packaging of DNA. So now let's understand how the packaging happens. So I've already told you DNA is negatively charged, right? And if it is negative charged, it can easily be wrapped around proteins that are positively charged. Yes? Yes or no?
Yes, sir.
So write down DNA is packed. DNA is negatively charged first. Write down DNA is negatively charged. And is packed with the help of positively charged proteins. Positively charged proteins known as histone proteins. Positively charged proteins known as histone proteins. You all did this mathematics. You all have written this calculation above.
Yes, sir.
Yeah. So histone proteins. Now let's see what are histone proteins. So there are different kinds of histone proteins. One kind is where 8 smaller subunits of the protein, 8. So DNA, when, let me draw it here. So there will be 8 smaller subunits that will come together to make a bigger subunit, which will be like spherical, like this. And this will be known as core histone molecules. And it is actually it is also known as histone octamer. Why it's called octamer? Because there are 8 monomers present here. Okay, if you divide it, it's 2, 4, 6, 8. You understand? There are 8 octamers that come together to make this histone. 8 monomers that come together to make this histone octamer. Is it clear?
Yes,
Yes,
sir.
sir.
Around it, the DNA winds. So I'm making the DNA in black. So DNA is this, goes here, comes back again, winds, and comes back. So you see the DNA is wound around approximately 2.5 times. So 1, 2.5. That is, if you calculate this length and this length also, it's like around 2.5 revolutions around the histone octamer. You understand?
Yes, sir.
And when here there is one more histone which is called H1 histone or histone 1. So this is like a clipper. Okay, you understood? So this is the core histone over which the DNA binds. Okay, around, make it as a double, it's double stranded, so it goes okay. And the histone one is like a clipper that clips here, keeps both these strands in place. Then again it goes and binds around the second histone, then the third histone, and it continues. You understand? So this DNA will go and bind around the second histone and third histone and fourth histone. And this kind of a structure, if you just see it under the microscope, then DNA will looks like this. So there will be histone proteins at regular intervals. Okay. And then there will be a long DNA thread will come, bind around it, and around it. Understand, everyone? And there will
Yes.
be clipper proteins that will clip the DNA. Okay. So if you see this structure, it will look like in a field where we are studying the DNA, it looks like this, that there is a thread and there is a bead. Yes, it will look something like this.
Yes, sir.
So this kind of structure is called beads on a string structure, because it looks like this only, right? And this kind of structure comes up because of this kind of a packaging of DNA, which is negatively charged over histone, which is positively charged. Is it clear?
Yes.
This is the first level of packaging, the first level to make beads on a string structure. Please draw this. Can you still see the screen? I just need to stop sharing the screen, people, for 1 second. I need to pull out a diagram. Yes. Okay. Can you all see the screen again that I'm sharing?
Yes, sir.
Yes. Drawn? This is done.
Yes, sir.
The first level of packaging to make beads on string structure. Now, let me show you how it. Can everyone see this diagram?
Yes, sir.
Yes. Now, pay attention to this. Understood? I will be using. So this is the DNA double helix. Yeah. So the 2 nanometer diameter is between the two strands. Okay. You understand? Between the two strands, it is 2 nanometer. Then this DNA is coiled around histones. Can you see histones here, everyone? The same thing that I told you here.
Yes, sir.
So this DNA binds around the histone, and then linker DNA is this part. This is linker DNA. The amount of DNA which is present between two nucleosomes. Oh, I forgot to tell. So this structure is called nucleosome. What it is called? Nucleosome structure. The first level of packaging. Is it clear? This is one nucleosome. This is one nucleosome, this
Yes,
is
sir.
second nucleosome, third nucleosome, fourth nucleosome, and together they form a bead on a string structure. So this linker DNA and these nucleosomes together form a chromatin. Okay. Chromatin is the bead on a string structure. So this is also known as chromatin. Chromatin. Is it clear? So chromatin is the second level of packaging. First level of packaging, so it starts from naked DNA, let's say. There is naked DNA to begin with. This is the naked DNA. Then the first level of packaging is nucleosome, okay, which is here. Then the second level of packaging is, which becomes your chromatin, okay, which you see here. Can you see this? The nucleosome together form a string together. That's called chromatin. Then the third level of packaging is chromatin fibers. So this chromatin again goes to form fiber. Fibers, one fiber, which is shown here. So can you see tight helical fiber? This one, everyone?
Yes, sir.
Yes, sir.
So this is the third level here. Okay. Then this chromatin fiber again coils and form a supercoiled structure, which is the fourth layer. This supercoiled structure is also known as chromatid, and it gives rise to chromosome. Is it clear? At the end. So can you see the levels of packaging that is done in the DNA?
Yes, sir.
Now here you can see that this is one pitch, right? And here you can see, look at this figure. Can you see that there are This is octamer, histone octamer. You see 1, 2, 3, 4, 5, 6, and there'll be 2 behind, around which the DNA is binding. So I have made this octamer as this one sphere, but you understood there are 8 little spheres which together come and make a octamer. Yes. And finally here
So,
it goes to form the chromosome that we know. Okay. Just to complete this figure. Understood, everyone?
yes, sir.
This is where the last layer comes. So this is how a very long DNA thread is supercoiled in the cell. So just write down this sequence. Levels of supercoiling. And remember this sequence. Done?
1 minute, sir.
Yeah. Done, people? Now, just remember this one more thing. Let's see, nucleosome, about the nucleosome, it is formed with histone proteins, right? DNA with histone proteins. So till this level, nucleosome and chromatin, it is with histone proteins, with the help of histone proteins, right? But this fiber and the chromatid, the chromosome, at this level, histone protein alone is not enough, right? Because it can just make bead on the string structure. But for further coiling, super coiling, and for further uh, binding, we need other proteins, correct? So from this stage onwards, we also depend on NHC proteins. Histones are no longer enough. So these NHC proteins are non-histone chromosomal proteins. Okay, which now makes a chromosome. Is it clear, everyone? Are you following? So in the early stages of packaging, we rely on histone proteins. But for later stages of packaging, histone proteins alone cannot be very helpful. So we need additional proteins which are called NHC, non-histone chromosomal proteins. They are not histone types, they are something else. Okay? Yes or no?
Yes, sir.
Yes, sir.
And to make a nucleosome, remember that around one histone octamer here, around one histone octamer, 200 base pair of DNA is wrapped. Okay. So on one octamer, around 200 base pair is wrapped to make one nucleosome. So how many pitches are there in one pitch? There was 10 base pair, right? So in 200 base pair, how many pitch? Approximately 20 pitches of DNA wrapped, correct? Understand or not?
Can you explain again?
Around one octamer. So this, from here, from this starting point to this end point, this DNA which is wrapped 2.5 times, okay, this is around 200 base pairs. You understand? On one nucleosome.
Yes, sir.
So on one nucleosome, this green ball that I have made, around 200 base pair, starting from 1 2 3 4 5 6, like this, 200 base pairs are wrapped. And I told you above, in one pitch, this one turn, how many base pair are there? 10. Yes.
Yes, sir.
So if they tell you that approximately 200 base pair of DNA is wrapped around one histone octamer to make one nucleosome, then around 20 pitches of DNA is wrapped, right? Because in one pitch there is 10. So in 200, so
Yes,
in
sir.
20 pitches there will be 200, 20 pitch of DNA. And can you also figure out the length of the DNA, exact length from this? Between two base pairs there is 0.34 nanometer. So 0.34 into 200. How many nanometers of DNA is present in one histone, in one nucleosome? The distance between two base pairs is 0.34 nanometer. 200 base pairs of DNA is wrapped on one histone octamer to make one nucleosome. So what is the total length of DNA present in one nucleosome? Simple mathematics. 0.34 into 200. What is the answer?
64 into 10 to the power minus 9 meters.
Which you can say 64 nanometers of DNA, right?
Yes, sir.
Yep. Everyone understood?
Yes, sir.
68 it must be, I think.
68, sir.
Yeah, right? 68 nanometer of DNA is present in one nucleus. These were just some neat questions, so I just thought of discussing it with you. Understood, everyone? The packaging. Everything written? Shall I move forward? Please say something, yes or no. You are,
Yes, sir.
yeah, okay. Because if you are writing, then I'll wait. It's done. Now, based on this packaging, sometimes some portions of the DNA are coiled or are packed very tightly. Okay, because this packaging is happening. So it is packed very, very tightly. It is packed so tightly that the central dogma could not like take place. Remember, I told you the central dogma above. DNA, from DNA, you need to make RNA, right? And now you realize that this DNA is packed, super coiled packed, right? So to make RNA out of it, something has to reach the DNA and then transcribe the bases and make a RNA, yes or no? Yes, everyone? If you don't understand, you said, please explain again.
Sir, can you repeat
silent.
again?
Yes, staying silent will not help. So let's say this is the DNA thread, okay? And you have to make a mRNA. So for that to happen, a protein has to reach this site, read the DNA from this side, this to this end, A to B, and then make a mRNA, right? And then this mRNA goes to make protein. You understand?
Yes, sir.
So to read the DNA, you have to reach the DNA. It's like
Yes.
you have to read page number 34 of a book. Now the book is in your hand. You will be easily able to read it, right? But if I make a pile of 10,000 books, let's say there are 10,000 books piled over one over the other in a room. Okay. And you have to read page number 34 of this book. Will it be easy? This red book. Will it be easy to open page number 34 of this book and read what is written on it? Yes, no, maybe, something. If something is very tightly packed one over the other, and you have to reach a small segment of that thing, is it easy or difficult?
Difficult, sir.
Difficult, right? Similarly, if the DNA is too tightly packed, it becomes difficult for the proteins in the cell or other factors in the cell to reach that part of the DNA or that gene. You know gene? By now you all know that gene is nothing but the segment of the DNA itself.
Yes, sir.
Yeah. So if one gene has to be transcribed, RNA from that gene has to be made. But that gene is present in the region where the DNA is so tightly packed that nothing can reach there. So do you think that gene can be transcribed easily? Can we
No.
reach out to that gene? No. So that gene acts like it becomes silent. No one can reach that gene and you cannot make protein from that gene. So based on the packaging, DNA regions are of two types. So write down. In a nucleus, some region of the chromatin, in the nucleus, some region of the chromatin are loosely packed. Okay. Whereas other regions of the chromatin are densely packed. Another analogy to make you understand is, if you are moving through a forest and if the trees in the forest are so densely grown like in a bamboo forest, will you be able to move through that forest easily? No, right? So if the DNA is loosely packed, and if I put a stain, like anything that colors that portion, so coloring will happen when the DNA will retain the stain, right? Stain is a pigment, right? So
Yes,
if that portion is loosely packed, will it retain the stain or stain will just move through it?
it will move
Loosely,
through it.
exactly, it is loosely packed, so it will not retain the stain. It will act like a sieve, and stain will just go through it, right? On the other hand, densely packed will retain the stain. So when you stain the chromosome, densely packed region is dark stained. Okay, whereas loosely packed region is lightly stained or light stained. You understand?
Yes, sir.
And this loosely packed region is called euchromatin. Euchromatin means true chromatin. And this densely packed region is called heterochromatin or heterochromatin. So euchromatin and heterochromatin. Heterochromatin is the densely packed DNA. Okay. Densely packed chromatin and loosely packed. Now, which of the following, which do you think will be easily accessible for transcription? So if I make a DNA, let me make a chromosome here. So if I make a chromosome like this, okay. So this is the chromosome. Okay. So some region will be, let me take a strain. This is one region that got stained, another stain. Understand? Some regions will be stained like this. Okay. Whereas the other regions will be stained lightly. Can you see the light stain also, everyone?
Yes, sir.
Now do you understand? It is the same stain that has been put. It's not different stains, but some region of the DNA retained the stain while the other region could not retain the stain. Yes?
Yes, sir.
Understood? So this densely, this lightly one is the euchromatin, right? And this dense one is the heterochromatin. You understand?
Yes, sir.
Yeah. Now, which one do you think is more available, easily available to transcription? Loosely packed or densely packed? Where the transcription will be easy to make RNA from the DNA?
Loosely packed.
Yes. So write down euchromatin. Euchromatin is transcriptionally active region, which means the gene present in euchromatin can be transcribed to form mRNA and to form protein. So the central dogma of biology can happen without any hindrance in a euchromatin, whereas in a heterochromatin, it's not possible. So write down heterochromatin is transcriptionally inactive region. Heterochromatin is transcriptionally inactive region. Is it clear?
Yes, sir.
Now, I have a question for you. Think about it and tell me. All the cells in our body, all the cells, they have the same genetic material. They have the same number of chromosomes, same genes, right? Yes or no? Reem, you there?
Yes, sir.
Yes, Reem. So all our cells in the body, they have the same genes, same number of chromosomes, or there is different genes and different chromosomes in different cells?
They have same.
Same. Okay. Then tell me, why do we have so many different types of cells in the body? We have a neuron, which is very, very different from a muscle cell. We have a fat cell where we store fat. We have a epithelial, squamous epithelial cell, which makes the lining of our organs. Then we have other types of cells in the body, eye, retina, photoreceptor cells. So how does all these different kinds of cells are made from the same genetic material, like from the same genes that are there in each cell? What tells a neuron to become a neuron and not a muscle cell? Is the question. What can you think? Can you think of something, Reem? You are right that all the cells have the same genetic material because our whole body is made up of is made from a single cell at one point of time, which comes, fuses, and make a zygote. Whatever is the genetic material in that zygote, every other cell from there onwards will have the same genetic material, correct? It's decided then
Yes,
and
sir.
there only. But during the course of embryogenesis, the zygote keeps dividing from 4-cell stage, 8-cell stage, blastocyst stage, which gets implanted. Then slowly different types of cells starts forming. So what do you think? How are we able to form hundreds of different types of cells having different function, different structure, present in different organ from the same information? Interesting question, no?
Yeah, sir.
Yeah. So that one thing that plays a major role is this. Through densely or loosely packing certain regions of the DNA, we can actually inactive or active certain genes in certain cell types at different time. We can regulate it. Okay, so let's say there is a gene which, if activates and if it starts producing mRNA and protein, it will make the cell into a neuron. So we have to silence that gene in a muscle cell, right? So that it does not become
Yes,
a
sir.
neuron, it becomes a muscle cell. Can you do it by heterochromatizing that gene? Can we do that? If that gene is heterochromatized, so it is not active now transcriptionally, so it will never form its protein. So it is true that we have around 28,000 genes in every cell of our body, but all these genes are not active and are not producing proteins in every cell of our body. Does it make sense to you?
Yes, sir.
Insulin as a gene, we have a gene for insulin in every cell of our body, but do you think our eye cells are producing insulin? Our muscle cells are producing insulin?
No.
Which organ is producing insulin? Pancreas. And in pancreas also, it is just the specific subcells that produce insulin, that act as endocrine gland. Do you understand? Everyone, is it making sense to you? Is it clear?
Yes, sir.
Yes.
Yep. Exactly. Okay. Well, good. We have the gene to produce melanin in every cell of our body. But we only produce melanin in skin cells when we are exposed to sunlight. Why don't our eye cells produce melanin? Or why don't our tongue produce melanin? Or the stomach produce melanin? Because it's not required there. You understand? So
Yes, sir.
that's how from the same information present in, it's just like the gene is the script of the movie. If we four, like Abia, Saifullah, Reem, Wafia, and me, we are 5 characters of that movie. So in that script, there will be dialogues written for each and every character, right? So all of us will have a full copy of that script. We should know what is the whole story, right? But
Yes,
what
sir.
part we will speak? Will we speak everyone's dialogue?
No.
No.
No, right? We will be only accessing certain regions and we will not be accessing certain regions. You understood the concept of euchromatin and heterochromatin?
Yes, sir.
Yes.
Okay, very well. Now, if you understood, so by now you have understood. I'm going slow because this is very important to begin with. You understood DNA, RNA, central dogma, structure, how it was found, the structure, what is the salient feature of the structure, then the packaging, how long is the DNA, how it is packed, what are the different layers of packaging, and after the packaging, how some parts can become euchromatin and other parts can become heterochromatin. All clear till now? No doubt?
No, sir.
Very well. Then let me go to the next section where we will study. Write down the experiments. Write down the experiments to figure out or to search for the genetic material. Now, today we know that DNA is the genetic material. And that's why it becomes easier for us to understand and learn certain things. But it was not the case, from always. Miescher, when he figured out that there is nuclein present in the nucleus and it is acidic in nature, and his proposition of he figured it out at around the same time when Mendel said that there are certain factors that are inherited from one organism to another or one cell to another. Okay, even both of these things we knew, but no one knew that what is that molecule inside the cell which is transferred. We know that something is transferred for sure. We know that it is present in the nucleus. Okay, we also know that it contains factors, like Mendel said. At that point of time we did not know about genes or alleles, but still we did not know that whether that molecule is a what is the nature of that molecule. Is it a lipid? Is it a protein? Is it a nucleic acid? Or is it something else? What it is? You understand? So basically no one knew what is a genetic material. Everyone knew that there is something which is a genetic material and it is inside the cell only. Do you understand the problem? People, do you understand?
Yes, sir. Yes.
So this is where I told you that chapter 6 goes into the details of chapter 5. So in chapter 5, whatever Sutton and Boveri did, whatever Thomas Hunt Morgan did, whatever Mendel did, they all proved that something is passing on, and those are factors, or you call it genes, or you call it alleles, and there is a linkage between the genes. Sometimes one gene governs one character, sometimes one gene can affect multiple characters, like in Pleiotropy. Sometimes multiple genes govern single character, which is polygenic inheritance. But basically, what is the nature of this genetic material was not known. So for this, there were many experiments that done. And the first in the series that we will study is called the first proof closer came from transforming principle by Griffith. So there was Frederick Griffith. Okay. Frederick Griffith in the late 1920s, he gave a transforming principle. He did an experiment and figured out transforming principle. Now, what is this? What does this mean? What was this experiment? So he was the first person to show that something passes on from one organism to another and changes the another organism physically. Okay, the physical form changes. Now this was very, very interesting because nothing of this sort was shown before. It was shown that something passes on to the offsprings. Mendel showed this, right? That something goes on from the parents to the offspring that decides how the offspring will look, how tall will they be, how other characters will be like, right? But Griffith showed that something can be sent from one organism to another without reproduction. Like you can just send it, you can just take it from here and put it there, and the other organism will change. That's what that was called transforming principle. Do you understand, everyone?
Yes, sir.
Yeah. So write down. Frederick Griffith used the bacteria. Frederick Griffith used the bacteria Streptococcus pneumoniae. I'm sure you all must have heard of this bacteria. Streptococcus pneumoniae. You know, what is this bacteria? Have you heard this name before? Pneumonia. This is the bacteria responsible for pneumonia disease. Hence the name. So the name of the disease was decided on the basis of the name of the bacteria. You know what is pneumonia? People, pneumonia. Am I audible?
Yes, sir.
Yes, no? Okay. You know pneumonia as a disease which affects the lungs? No? Okay, figure out pneumonia. Okay. Now, this bacteria has two strains. Okay. Strain 1 is called smooth strain. Okay, also known as S strain. Because this strain produced smooth colonies, shiny colonies. Okay. Produced smooth and shiny colonies on a culture plate. Okay. And this strain is lethal. Okay, or virulent. Lethal strain. The second strain of this bacteria was strain 2. It was called rough strain or R strain for the word rough. And this strain produced rough colonies. They were not shiny. Is it clear, everyone? Till here, you are understanding?
Yes, sir.
And they were non-lethal. Right? Infection with this strain will not kill a person. Okay. But infection with S strain will kill. Okay. You understood both the strains?
Yes, sir.
Now this was the experiment. Let's see what was the experiment. Now you tell me the results, okay? First, he took S strain, Griffith took the S strain and injected it in the mouse. What do you think will happen? S strain
It
is
will die.
lethal strain. Yes. Simply mice die. Right? Makes sense. It was expected also because it is S strain, it is lethal. Right?
Yes, sir.
Then he took the R strain and did the same, injected in the mice. What do you expect now?
He will live.
The mice lives. Expected, right?
Yes.
Because it's a non-lethal strain. Now this is not interesting bit. Interesting bit is he took S strain, but before injecting, he heat killed. Heat killed means if you boil a bacteria, the bacteria dies, right? That's why we boil water to decontaminate any water, right?
Yes,
Yes.
sir.
So if you heat kill the strain and then you inject it in the mice, what do you expect? Mouse will live or die? You have heat killed the bacteria, heat inactivated the bacteria.
It will live.
The mice will live. Now, if the S strain is put without heat killing, the mice will die. If we heat killed, which means that now the bacteria is unable to kill the mice because the bacteria is dead. Yes? Yes or no?
Yes.
But that's not all. The most interesting thing was where he took the same heat killed S strain, which was heat killed. So if you put S strain alone, the mice will live. He added this S strain with R strain, which was live, not heat killed. So if you inject S strain alone, the mice will live. If you inject R strain alone, the mice will live. In both the cases, mice should live, right? But when you mixed both the strains and injected in the mouse, what do you think must have happened? The mouse individually, this could not kill the mice, this could not kill the mice, right? This did not. See here. This also lived and this also lived. But when you put both of them together here, do you understand what is being done now? Everyone.
Yes, sir.
Two things which alone could not kill the mice, now when injected together, the mice die. This was the most interesting thing. How could the mice die when we have injected S strain which was heat killed and a R strain which anyway was not able to kill the mice? Then why did the mice die? And not only this, then from the dead mice, he actually could recover the live S strain bacteria. He did not injected any live S strain. Then how could he recover live S strain from the bacteria? These two results actually is because something from the, so the S strain is heat killed, so the bacteria is killed, that's true. But the genetic material of the bacteria is not degraded, all of it. You understand? It's well protected inside the nucleus. So that genetic material is still intact, and the transfer of genetic material from S strain to R strain must have happened, which transformed this R strain into a live S strain, which killed the mice. You understand, everyone? So there was some gene which were only present in the S strain, and they produced toxins that could kill. R strain did not had that gene, let's say. Now, after we killed the S strain, the DNA is available. The DNA is just there. And when you mix both of them, some part of the DNA from the S strain can be taken up by the R strain. This thing bacteria can do. Bacteria can actually take up random DNA from surroundings and incorporate it into their own genome. And because of which the R strain got transformed into the S strain and could kill the mice. Do you understood?
Yes, sir.
So write down the conclusion. I'm dictating right down. Griffith concluded that the R strain bacteria was somehow transformed. The R strain bacteria was somehow transformed by the heat killed S strain bacteria. By the heat killed S strain bacteria. This must be due to the transfer of the genetic material. This must be due to the transfer of genetic material but the biochemical nature of the genetic material was not, but the biochemical nature. What was the genetic material? He proved that something is the genetic material which, if you can transfer from one person to another or one bacteria to another, can change the physical appearance or properties of the other person or the other bacteria. But the biochemical nature of the genetic material was not defined from this experiment. What he achieved was, which was not achieved before. Everyone thought genetic material can only be transferred from parents to the offspring, but he showed that it can also be transferred from one cell to another and can transform the other cell. Do you understand, everyone? The transforming principle is clear. Saifullah, Wafia, and Abia.
Yes, sir.
Yes.
So tomorrow from next class, we will be now learning about how did the biochemical nature was figured out. From this experiment, how did we move forward? Okay. So this is where I end today. If you have any doubts, please ask. Otherwise, I'll see you tomorrow. Good night, everyone. Take care. Any doubts? No doubts? Okay, then.